We have had a look at the basics of gene regulation in prokaryotes first, and then in eukaryotes. Let's take a look at how chromosomes are organized and how chromosome structure influences gene expression. So we're going to talk about DNA and chromosomes. Now let's take a look at the different forms of chromatin that you can see in a nucleus. We recognize heterochromatin and euchromatin. The darker granulated stuff in a nucleus is typically referred to as heterochromatin, and the lighter granular stuff inside a nucleus is euchromatin. There are two grayish areas in this particular nucleus, in this cross-section, and those are the nuclei, which are built around a portion of DNA that encodes ribosomal RNAs, and so nuclei are the source of ribosomal RNA synthesis, in particular 28S and 18S and 4S RNA. Euchromatin is less condensed, heterochromatin is more condensed. So let's imagine heterochromatin as being coiled in this cartoon and euchromatin as being not coiled or less coiled. It is reasonable to suppose that euchromatin is more accessible to the enzymes like RNA polymerase that are necessary to do transcription. Therefore, it's also reasonable to suppose that euchromatin is more rapidly digested by DNase than is heterochromatin. Let's go back and take a quick look. So there's our cartoon of coiled and less coiled chromatin, heterochromatin and euchromatin. The less coiled material, the euchromatin, should be attacked by DNase more readily and more quickly than the coiled material, the, say, solenoid or higher order structure of chromatin. Because being more condensed, heterochromatin would be resistant to DNase digestion. Now, I'll catch up with myself. It could be argued that DNA that is actively being transcribed, DNA whose genes are actively being transcribed, are more likely to be in euchromatin because if euchromatin is more accessible to DNase in a test tube, then in the nucleus it should be more accessible to the enzymes of transcription, namely RNA polymerase and associated factors that are necessary to get that to happen. Here's transcription occurring in euchromatin, but not in heterochromatin. You would cartoon it in this way, and you would be showing nascent transcripts. So go back and look, and you can actually see the lamp brush chromosomes, which have this feature. But right now, it's just a cartoon to drive home the message that euchromatin might be more active in this hypothesis than heterochromatin, more active in transcription, and so you would expect nascent transcripts, right? Well, we can test with deoxyribonuclease whether euchromatin contains active genes versus heterochromatin containing inactive genes. Here's an experiment. Birds' red blood cells have nuclei, unlike our erythrocytes. Our erythrocytes were produced by cells that originated in the bone marrow. They are called reticulocytes. But eventually, after the reticulocytes produce the hemoglobin and other components of the red blood cell, the nucleus is destroyed, basically. So our red blood cells look like these donut-shaped things, and they don't have a nucleus, and they function for about 60 days before the spleen takes them out of commission. But birds have nucleated red blood cells. So if you take chick red blood cells from young chickens and isolate their nuclei and do a chromatin extract that yields total chromatin in a test tube, cartooned again to suggest some of it is heterochromatin, some of it is euchromatin. If you now digest this preparation with DNase for different amounts of time, the expectation again is that the euchromatin will be digested preferentially and the heterochromatin will remain behind. So what if you were to now extract undigested DNA from such a digest? Well, the way in which this experiment was actually done was that the chromatin extract was digested with DNase, again, for different times, not just completely. So what you do is you set up a test tube, you put in the chromatin and the DNase, and after different times you pull out some of the extract, the digest extract, and you extract the DNA from each of those tubes that you have pulled out at time 1, time 2, time 3, and so on. You extract the DNA from each of those test tubes, and you put a little spot of DNA in a different position on a filter shown here. So each circle represents a small drop of DNA extracted from progressively longer digested bits of red blood cell chromatin. So on the far right would be the DNA taken from the most digested chromatin. On the far left, DNA from the least digested chromatin. You make a duplicate of this filter. So you have two of these filters, and you'll see why in just a moment. The next step requires a moment of explanation. We're going to ask whether we can detect a gene for globin, the protein that is part of hemoglobin, whether we can detect the gene for globin in the chromatin digests from a red blood cell. So we need to do that by using a cloned gene, a cloned globin gene, as a probe. That means we're probing for globin genes using a known DNA sequence that we know codes for globin. And why would we do that? Well, first of all, you tag the globin so that it's now radioactive. That's what the little red asterisk is supposed to tell you. So we have radioactive probe, which means we have a radioactive globin gene. What have we done? We've denatured that gene, so we have single strands. On the filter, we actually have placed the DNA in a single-stranded state. And what the probe will do, what the denatured radioactive globin gene DNA will now do, will find sequences complementary to each of the two strands in the probe and form H bonds, even on the filter, leaving then a deposit of radioactive material on the filter, which can then be detected. So we allow the probe DNA, this radioactive DNA, after denaturing, to, we say, hybridize. We mean hybridizing in the sense of if the two original parent strands of DNA were artificially separated and we introduce two new strands that are potentially complementary, and they do form H bonds, well, they've kind of made a hybrid DNA structure. So we say we're going to allow the radioactive probe DNA to hybridize to complementary sequences on the filters if they're there. And then we have to expose and develop an autoradiograph, which will tell us which of the spots is radioactive and how radioactive are the spots. So here we have our two filters. You don't see them at the moment, but they have the spots of DNA on them. What you're really looking at now are the pieces of film that have been placed over the two filters. And again, to orient you, the increasing time of digestion of the chromatin initially is from left to right, and the DNA has come from red blood cell chromatin that has been digested with DNase for different periods of time, progressively towards the spots on the left. Now, if you expose and then develop the autoradiographs, this is what you're going to see. On the upper filter, we have probed it with the globin gene probe. And look what happened. The spot on the left shows the largest amount of radioactivity, which means the largest amount of globin gene in the DNA. That's the least digested. But as you look towards the right, the amount of probe that can bind to the DNA in the spots progressively more to the right goes down until you reach the relatively small amount shown at the spot on the far right. Why is that? That's because DNase digestion for a longer time has managed to digest most of the euchromatin, which we are proposing is the location of active genes, such as the globin gene in a red blood cell. Well, here's the control. Chickens make ovalbumin, which is nothing more than egg white protein. That's made in the oviduct, not in a red blood cell. So you would assume that the ovalbumin gene in a red blood cell is in heterochromatin because it's not expressed in a red blood cell, and so it should be packed up and put away, so to speak. And so what you would expect then is that no matter how long you digest red blood cell chromatin, there will always be a fair amount of ovalbumin gene left behind in the DNA after the digestion. And that's exactly what you see. I don't have a slide to show you, but I can tell you that when this experiment is done in the inverse, if you will, you get the expected results. Now, what do I mean? I mean if you do the same experiment, except this time you extract chromatin from oviduct tissue, make your digests and make your filters, and then probe with the globin gene probe or the ovalbumin gene probe. This time, it's the ovalbumin gene that disappears very quickly over the time of digestion, whereas the globin gene remains roughly at the same amount in each of the digests of oviduct tissue chromatin. So the conclusions, of course, are that the ovalbumin gene in the red blood cell chromatin is resistant to DNase digestion, and that's because it is a part of heterochromatin rather than euchromatin, And likewise, the globin gene DNA, which gets digested very quickly after treatment with DNase 1 of chromatin from a red blood cell, the globin gene disappears quickly because it's active and it's in a region of chromatin that is more open, in a more open configuration, and therefore more accessible to DNase, but also in the cell, again, more accessible to the enzymes of transcription. So that brings us to the end of this presentation, which was intended to both show you the structure of chromosomes and therefore what any kind of DNA manipulation has to contend with. The cell has to deal with DNA wrapped up in proteins in the first instance to do anything, whether it's express a gene or gather components together to do mitosis or to silence a gene. And we've actually demonstrated that chromatin really does come in two flavors, euchromatin and heterochromatin, and that the euchromatin, the looser packing, is the site of active genes.